The purpose of this video is to talk about the basic principles of vaccination and to share with you an overview of the design of current vaccine candidates for COVID-19. To also discuss some of the concerns and misperceptions around these vaccines. Coronaviruses like SARS-CoV-2 that cause COVID-19 are single-stranded RNA viruses. Their RNA genome is encapsulated in a membrane that is studded with surface proteins. The virus makes about 27 different viral proteins, the major one being a protein on the surface called spike protein or S protein. And that's shown down here on this image of the virus. When the virus enters our bodies, it is able to be recognized by a receptor on our cells called ACE2, and this is how it gains entry into the cells. Then once inside the cells, it hijacks the machinery inside our cells to produce more viral particles. These viral particles are then released from the cell and they continue the infection process. Now, all viruses, including the one causing COVID-19, are recognized by our immune system, and our immune system mounts a response that will help limit infection. When the virus enters, it is presented to cells within our immune system, and this leads to two responses. One, a T-cell mediated response that identifies and degrades infected cells, as well as an antibody mediated response. These antibodies, including the ones we really want, which are called neutralizing antibodies, will bind and block the virus from infecting any new cells, thereby limiting the infection. Vaccines are really just designed to trigger this immune response before you see the real virus. Many of the traditional vaccines were just weakened or inactivated versions of the virus itself. This protection that you might receive from a vaccine could last many years or even a lifetime. We have examples from measles, mumps, and rubella as that being the case. Or they can also just be seasonal, like influenza. The seasonal nature of the flu vaccines is really caused by the fact that these are rapidly evolving or mutating. And we also have two surface proteins to factor in with this virus, the H and the N. And those that are developing vaccines for the flu need to get the H and the N correct in order to have maximal protection from the flu each season. One interesting feature with SARS-CoV-2 is that you don't need the whole virus in order to produce an effective vaccine. And that's really the first key point that I want to make, is that only the spike protein, that protein on the surface of the virus, is necessary to generate a suitable vaccine. We know this because the spike protein is the primary target of a vast majority of the antibodies that are found in infected patients. So people have set about using two different platforms, classical as well as next generation platforms, to develop a vaccine based on this knowledge. The classical platforms are really just using the whole inactivated or weakened virus, or using purified protein subunits as vaccines. But there's a whole new set of platforms. These are the next generation platforms, and these are the ones that involve the use of mRNA or messenger RNA that's bound in lipid particles, or using different viruses as sort of a delivery system for the information that you need to make the spike protein and cause an immune response. The second key point I want to make is that all the next generation platforms have the same end goal. They are aimed at delivering the genetic material, whether it's DNA or RNA, for you to make the spike protein so that your immune system can create that protective response when you run into the real virus. The last point, the third point, is that while these are new platforms, they are not new biology. They take advantage of nature's own mechanisms to induce immunity. Instead of providing the virus or the protein subunit, we're simply providing the genetic material to make that spike protein and create the same end response. This slide provides an overview of these platforms. On the left are some of the classical platforms, which are, again, whole inactivated virus. We have an example prior with polio vaccine, live weakened virus or attenuated virus, which is this measles, mumps, and rubella, and then the protein subunits, which are used for the flu vaccine. On the right side here is an overview of the next generation platforms. These can be DNA or RNA that is encapsulated in these lipid nanoparticles, or they can be that same material encapsulated in a different virus, and that's what's used as a delivery system. So as of September, there was almost 300 different vaccine candidates under exploratory and preclinical development, as well as around 25 or 30 that were in various phases of clinical trials. And those include ones from Moderna and Pfizer, which are mRNA-based, and AstraZeneca, which is a viral vector-based vaccine, that were in late phase and are now approved for emergency use and will be approved for full use shortly. So talk a little bit more about the messenger RNA, or mRNA vaccines, and these again are from Moderna and Pfizer. The vaccine consists of the messenger RNA that encodes the spike protein, and it's encapsulated in a lipid-based package called a nanoparticle. Once this is delivered, the mRNA is released from the nanoparticle, and your cells use that mRNA to make the spike protein. That spike protein is then recognized by your immune system and triggers this immune response and gives you the protection that you need. One important feature with these mRNAs is they are not designed to integrate into our genome. This is a very unlikely event. It would first need to be converted to DNA. That DNA would need to be sent to the nucleus, and then it would need specific sequences which it lacks in order to integrate into our genome. None of that is present in these vaccines. In fact, when the mRNA enters the cell, it's almost immediately going to be recognized by the ribosome and used to make the spike protein. So there would be almost no opportunity for that process of converting to DNA and integrating to take place. One big benefit of these vaccines is this type of vaccine is that they can be made quickly. So you can make a new one if the virus were to mutate by simply changing the mRNA sequence. Both of the mRNA vaccines have been shown to be highly effective, greater than 90% in initial trials. I'll talk just briefly about this one from AstraZeneca. This is the use of a non-replicating viral vector delivery system. It uses the coat of another virus, which is an adenovirus, similar to the one that causes a common cold, to deliver the genetic information that you need to make the spike protein. So in this case, again, you're just using a virus as a delivery system. It's delivering the genetic material to make the spike protein and give you this immune response that provides immunity. These are non-replicating viral vectors, so they are a one-time delivery system. The virus itself, the adenovirus, will not replicate within your body. One other interesting feature is instead of using a human adenovirus vector, these use one from chimpanzees. And that's because in many older people have already seen the human adenoviruses, and therefore the vaccine would be less effective because our immune system would recognize this and clear those vaccines based on the prior immune response that you have. So these were 62% effective with full 2 doses, but again reached that 90% plus effectiveness if you first gave a half dose and then the full dose for the second one. A little bit more about stability and dosing. So the Pfizer vaccine requires very low storage temperatures, that's below 80 degrees Celsius, while the Moderna vaccine only needs minus 20 degrees Celsius, like the freezers we have at home. This is really due to the fact that they have a different composition to their lipid nanoparticle, and Moderna's composition makes the mRNA more stable at that temperature of minus 20. The AstraZeneca vaccine remains viable and effective at only 4 degrees, so refrigeration. This is a big advantage for delivering this vaccine to different locations around the world that lack the capability to store at lower temperatures. All these current vaccine candidates require multiple doses. The first one to prime the immune system, and the second one is boosted to give you a full immune response. And they have different cost structures as well. Just to touch on some of the common questions that come up around safety and other concerns. One is, will we need a new vaccine every year like the flu? This isn't fully known, but the thought is that we will be able to get at least a couple years immunity from this current vaccine. One other question is, in the initial trials, have there been any serious adverse events? There are very few, if any, serious adverse events associated with these initial clinical trials. There have been some side effects, including pain at the injection site and other symptoms that have really been fairly transient and only last either half a day or maybe a full day before the person is fine. One other concern that's raised is won't the compressed timeline for vaccine development compromise the safety? The point here, I think the reason it's been moving so quickly is not because the developers are cutting corners in terms of safety. What they're doing is increasing process efficiencies. They're not waiting for each step to be confirmed before they go on and take steps to gain approval and monitor the benefit at later steps. So not doing things serially, but instead in a parallel fashion is really what's driven the speed of this vaccine development. And then lastly, for those that might think, why do we need a vaccine? This is really our end game. This is our opportunity to get life back to normal. We have two choices with a pandemic. It's either you let the virus run through and infect people naturally, and that's how we generate immunity, or we do a vaccination program. And I think for this virus, it's particularly important that we move forward with this vaccination program so that we don't overwhelm our healthcare system. So I thank you for your attention and wish you all the best holiday season.